My name is Sabrina Spencer, and I'm an assistant professor in the Department of Biochemistry at the University of Colorado Boulder. I received the Beckman Young Investigator Award in 2016. Broadly, my research is focused on understanding the causes and consequences of non-genetic heterogeneity. That is, trying to understand why even genetically identical individuals don't always behave identically. Let's first take the macro world example of my identical twin sons, who, by the way, have been with me since the beginning of my Beckman adventure after interviewing with me for the award in utero. So let's interview them to see what they have to offer on this subject.
Boys, can you introduce yourselves?
No. Yes. We are identical twins.
And what are your names?
Sebastian and Julian.
And do you like all identical things?
No.
Can you give an example?
I love olives.
Saba, do you like olives?
Nope.
And what do you like?
Cantaloupe.
Julian, do you like cantaloupe?
No.
Through the Beckman Young Investigator Award, we have developed methods to quantify the impact of cellular heterogeneity in biological systems, which has applications ranging from basic science to the clinic. We have focused on the molecular origins of drug resistance in cancer, where a few outlier cells that manage to evade treatment can seed a population that drives acquisition of drug resistance mutations, leading to tumor relapse. Sometimes outlier cells arise due to genetic differences between cancer cells, but drug-tolerant cancer cells appear even when using a clonal population of cells. This rapid, non-genetic adaptation to cancer therapeutics is increasingly understood to be a widespread problem in cancer therapeutics, but is still very poorly understood. We chose melanoma, a deadly form of skin cancer, as our model system since we had strong preliminary data showing heterogeneity in drug response. On the left, you can see a population of drug-treated melanoma cells marked in blue, with a pink overlay on cells that are actively proliferating. While the drug treatment on the right does block proliferation in the vast majority of cells, there are always a few that are still dividing, no matter how high of a drug dose we use. How rapidly does this occur, and in what fraction of the population? And are these rapidly escaping cells really the source of drug resistance? The answers to these key questions have been hindered by a lack of technology to follow drug responses in single living cells over time. To understand where these proliferating cells are coming from, we had to develop new methods to track them over time. Through the Beckman Young Investigator Award, we have done just that. Our new cell tracking pipeline, called ElipTrack, merges the best features of existing cell trackers and adds several new features of its own, yielding the best performing cell tracker for long-term tracking of hard-to-track cells, such as cancer cells. Each nucleus is fitted with an ellipse and tracked through multiple generations, enabling us to generate cell lineages, as shown on the right, where heterogeneous behaviors can be easily detected. Let's look at a few key results from our Beckman-funded project. Applying this cell tracker to a movie of melanoma cells expressing a CDK2 activity sensor as a marker of proliferation, we can visualize two divergent behaviors upon treatment with drug. Both cells divide around the same time and then enter a 2-day long quiescence where they are not dividing. But then, the cell with the orange arrow somehow manages to overcome the drug, building up CDK2 activity and dividing, while the well-behaved blue cell remains quiescent. Is this just a flash in the pan, or does the orange cell continue to have increased proliferation potential over extended drug treatment? To answer this question, we filmed and tracked melanoma cells over a 10-day drug treatment, and looked to see if the cells that escape drug in the first 4 days tend to outproliferate non-escapees during the rest of the movie. Each line in this heat map is a single cell's CDK2 activity, where red means high activity. And it's clear that cells that rapidly rewire to escape drug treatment early in the treatment, those are the cells in the top half of the plot marked escapees, outproliferate the non-escapees, as seen here, and have 4 times as many mitoses in the final 6 days of the movie. Unexpected finding was that cells that attempt to cycle in the presence of drug incur increased DNA damage relative to untreated cycling cells and relative to non-escapees under drug treatment. This can be seen by the overlap of the green DNA damage marker with the pink proliferation marker in the bottom right image. In summary, we have developed ELIPTRAC, a new cell tracking pipeline for long-term tracking of time-lapse microscopy movies. Using these new methods, we found that a subset of melanoma cells can rapidly rewire their proliferation programs to escape MAP kinase pathway inhibition. Cells that escape drug and enter the cell cycle experience heightened DNA replication stress and DNA damage, yet outproliferate non-escapees. Thus, these early drug escapees may be the seed population that enables eventual drug resistance, and if eliminated, could reduce tumor relapse. 2 manuscripts describing this work are now posted to bioRxiv, and ELIPTRAC will soon be published in Cell Reports with all of our code made freely available. Time and again, the development of new methods has led to new discoveries, as Dr. Beckman's development of the pH meter and other inventions have so clearly shown. In the fields of cell biology and cancer biology, reliable cell tracking has been a major bottleneck since the dawn of time-lapse microscopy. Our new experimental and computational methods for long-term time-lapse microscopy and cell tracking set the scene for many potential future discoveries in these fields. Indeed, ELIPTRAC is now powering discoveries outside of our Beckman-funded project and was an essential component of my lab's paper in Science out this month. Our Beckman-funded work also opens several burning questions in the cancer biology realm. First, how do cells rewire their proliferation pathways to override drug blockade and re-enter the cell cycle? Our Beckman work has a few promising leads, but more work is needed to draw firm conclusions. Second, why do cells that manage to cycle in the presence of targeted cancer therapeutics incur DNA damage when these drugs are not inherently mutagenic? Third, how widespread across different cancers and different drugs are these phenomena of rapid drug escape and the associated DNA damage? Most broadly, similar to how the discovery of induced pluripotent stem cells showed that cells are much more plastic than previously thought, I believe our methods can be applied broadly across biology to answer fundamental questions about the extent of non-genetic plasticity and heterogeneity of living systems. These questions form the topics of grants I am currently writing to continue the work that the Beckman Foundation funded early on. In closing, I would like to acknowledge my lab, the contributors to our Beckman project in Boulder, and of course the Beckman Foundation for selecting me for this prestigious award, which helped me propel our work in a new direction.
Thank you for